Okay, so now that you've looked at all these cancer cards, let's sort of look at some trends. So I'm going to show you on your homework assignment, just looking at this group of cards, one of the questions it asks you is what type of mutation is most likely or most often occurring. And you can see in the types it's either a tumor suppressor. Now remember, a tumor suppressor is an off switch. It turns the cell cycle off. We'll pull up the cell cycle here. All right, so we're going through the cell cycle. It would be one of these stop signs. It would say, nope, you shouldn't go on. That's a tumor suppressor. An oncogene is normally a proto-oncogene, which would turn the cell cycle on. But when it gets turned into a cancer cell, it's turning, or a cancer gene, it's turning the cell cycle on over and over and over again. So it'd be something that would cause the cell cycle to blow through the stop sign. Okay, so why would tumor suppressors be more common than proto-oncogenes? And that's because of dominance and recessiveness. So let's think about this for a minute. A tumor suppressor that turns the cell cycle off, right? You have let's say you have two alleles for that tumor suppressor and one of them gets wiped out so it doesn't turn it off. But you have another one that'll turn it off. So in order to get cancer from a tumor suppressor, you'd have to wipe out both of them, meaning you'd have to be homozygous recessive, right? For a proto-oncogene, if you turn one into an oncogene, so it's turning the cell cycle on, it doesn't matter if you turn the other one on, right? All you have to do is turn one on. So it's like a dominant mutation. If you turn one on, it's going to rush it through the cell cycle over and over and over again, regardless of what the other one is saying. Okay, and so oncogenes are usually dominant mutations and tumor suppressors are recessive mutations. And so you can see, like in this patient that has hepatic cancer, they had several mutations in a tumor suppressor because they had to be homozygous, recessive for all of those mutations before they ended up with cancer. Whereas if you just wipe out one oncogene, you're likely, or overactivate one oncogene, you're likely to get cancer. So you see the oncogene showing up only in one or maybe two, whereas the tumor suppressors, you have to get a lot of mutations for it to usually cause cancer. The other one asks what is the least common of the functions. So functions, again, you have the cell cycle, whether it's showing it pushing it through the cell cycle. The cell fate is what will the cell become, and then a genome maintenance, which is what repairs DNA. And you see that genome maintenance is the least frequent mutation. There's a lot of reasons this could be. This one's not as straightforward. Genome maintenance, it could be that any kind of mutation in a genome maintenance actually leads to cell death, programmed cell death, rather than cancer because it's just not survivable because they're that important. It could also be that those particular genes are in an area of the DNA that's less likely to mutate. It could also be that if you get a genome maintenance mutation, there's a lot of backups to that gene, right? We've got lots of copies of genome maintenance, so you can wipe one out and it doesn't necessarily lead to cancer. There's a lot of reasons it could be, but that is one of the things you should have noticed is that genome maintenance mutations are the least frequent. Maybe the other ones, cell fate and cell cycle, are more likely to cause cancer. So that's that one.